So chapter 16 is entitled The Molecular Basis of Inheritance. Watson and Crick are well known for having introduced the double helix model to describe DNA structure back in 1953. All of the hereditary information that is passed from one generation to the next is encoded in that DNA and is found in all cells throughout the body. And that DNA is able to produce proteins, which then can direct the development of various traits, including those that are biochemical based, anatomical, physiological, and to a certain extent behavioral. So this is kind of relating back to what we were talking about with chromosomal inheritance in chapter 15. This is prior to DNA being identified early in the 20th century. Identification of molecules inheritance loomed as a major challenge to biologists. Morgan's group was able to show that the genes were located on chromosomes, and there were two components making up chromosomes, DNA and protein. Don't know why that's showing up, but okay. And so those two macromolecules became candidates for the genetic material. And so in order to determine which was the genetic material, it depended on the organisms that were being used to explore those possibilities. And so DNA's role in heredity was first able to be discovered by examining bacteria and viruses that infect bacteria. So DNA can transform bacteria was discovered by Griffith. Griffith worked with a pathogenic or virulent strain and an avirulent strain that was harmless. And he mixed heat-killed remains of the pathogenic strain with living cells of a harmless strain. And it caused those living cells to be transformed and instead of being harmless, being harmful. That transformation is what he described that phenomenon as, which was basically a change in your genotype or phenotype due to the assimilation taking in foreign DNA. So kind of took this a step further and put these cells into mice. You had the S cells, the avirulent strand, the virulent strand that would cause the mouse to die. Then you had the avirulent strand that caused the mouse to stay healthy. Then he took the cells that were virulent and heat killed them, and the mouse lived. But when he mixed the heat killed cells with the living avirulent cells, the mouse died as a result of those R cells being transformed into S cells. So that was just the idea of transformation. Now, Avery, McCarty, and McLeod were able to identify DNA as being the transforming substance. And there was still a lot of debate about whether that was truly it because there wasn't a whole lot known about DNA at the time. Hershey and Chase were able to describe that viral DNA could cause changes in cells. When viruses are able to infect bacteria, they're known as bacteriophages or phage, and they're used a lot in molecular genetics. If you decide to do any research in college, you'll probably run into these. They did some experiments to determine that DNA was the genetic material of T2. Viruses have both a nucleic acid and a protein, and so they determined which of those components was entering the E. coli cell, and they determined it was the DNA inside of it as opposed to the protein that was entering and therefore passing on that genetic information. And they did this with a couple of radioactive isotopes. So we've got radioactive sulfur, which is going to cause the protein to become radioactive. They give it an opportunity to enter the cell, and then they blended all of the phage DNA together, and they see that the radioactive protein was in the liquid and that the pellets contained the bacterial cells, so it had not entered the bacterial cells. Remember that there's a phosphate group that's part of your nucleic acids, and so when the DNA was labeled with this radioactive isotope and blended together. Now we see that the radioactive component is in the pellet, which would be where the bacterial cells are, hence why it was DNA as opposed to protein. Chargraff you definitely heard a lot of before. DNA is a genetic material. We talked previously about what makes up DNA. DNA is a polymer of nucleotides, that they consist of 3 components to your nucleotide: nitrogenous base, a sugar, and a phosphate group. Chargraff was able to report in 1950 that the composition of DNA will vary from one species to the next, and that kind of helps to explain why it would be a good candidate for genetic material. Now, when he says that the composition varies, he's not saying the structure of DNA varies, but that those bases can vary. And so we knew that there had to be differences in the DNA if we're going to have all these different species that are similar but not exactly the same. So, as opposed to just having your genus, that we have all these individual species. And so, Chargaff's rule was that the base composition of DNA is going to vary between those species, and that regardless of what species you're working with, the amount of adenine and thymine bases you have are equal, and the amount of guanine and cytosine bases are equal. And there really wasn't a whole lot of awareness at this point as to why that was so essential that you have equal amounts of A and T and G and C. But when the double helix was discovered, that became more clear. So there's your basic nucleotides, and they have been joined together. So you have your polymer of polynucleotide, and in this case it's DNA because of the specific sugar, deoxyribose. I do want to point out the 5' and 3' end on this slide because that is going to come into play a little bit when we talk about replication, again, transcription and translation. The 5' end would be the carbon that is bonded to the phosphate group. And if you label your carbons, we did that earlier this year, going clockwise, 1, 2, 3, 4, 5, you'll see that the 5th carbon is bonded to the phosphate group, and that the 3rd carbon is bonded to the hydroxyl group. And it's that hydroxyl group that would have to give up a proton along with the other nucleotide that's coming along is going to lose a hydroxyl group so that they can form a covalent bond so that they will become connected to one another. They'll be part of the nucleotide or the polynucleotide. And so you can only make that connection on the 3' end of the sugar because that's where you have your hydroxyl group that's free and open. You cannot make that connection on the phosphate end, the 5' end. Wilkins and Franklin performed X-ray crystallography to examine the structure of different molecules. She did this with DNA and was able to capture an image as shown below that allowed Watson to determine that DNA had to be helical. And because of these pictures, he was able to kind of predict what he thought the width of the helix would be and how the nitrogenous bases would be spaced. And because of the way the picture portrayed the DNA molecule, it made more sense that it existed in the form of a double helix. So that's what we have now, and the 2 bases that are running on the anti-parallel strands making up your double helix are hydrogen bonded to one another. 2 hydrogen bonds between the adenine and thymine, 3 with the guanine and cytosine. So we have this idea of a double helix. We have the bases, and this all makes sense with what we've seen from the chemistry of DNA as well as the X-rays. Franklin had determined based on her crystallographs that there was 2 sugar phosphate backbones, with the nitrogenous bases being paired in the middle. And so Watson's model accounted for the fact that they were running in opposite directions. The DNA had to have, initially they thought it had to have the adenines together, the thymines, the guanines, and the cytosines, but that didn't make sense spacing-wise. So if the 2 strands truly were identical to one another, they would have to be set up in a complementary fashion, which is why adenine would have to go with thymine and guanine would have to go with cytosine, which makes sense with Chargraff's rules. If you had two purines paired together, the spacing between the backbones was too large. And if you only had the two pyrimidines together, which is what you would have had if the bases had been identical, then it would have been too small. But you needed that blend of a purine with a pyrimidine to be consistent across the board for it to make sense with the pattern that was predicted from the X-ray crystallograph. And so they determined from their work that, again, adenine is always going to go with thymine, guanine is always going to go with cytosine. There you can see where those hydrogen bonds are forming. Remember, hydrogen bonds form with an electronegative atom like oxygen or fluorine or nitrogen, and they are able to hydrogen bond with hydrogens that are bonded to adjacent molecules that are attached to one of those electronegative atoms. So the whole process of DNA replication and repair is kind of based on this double helix structure that was described by Watson and Crick with massive assistance from, my goodness, I can't believe I'm forgetting. Sorry, it's been a long day. Franklin, let's go with that one. They thought that the way that the double helix was structured perhaps suggested a way in which it could be copied and replicated. Because the strands were complementary, you could use each strand to build a new strand. And so what we know now is that through DNA replication, the parent molecule will unwind and daughter strands get built on those parent strands based on our base pairing rules, adenines with thymines, guanines with cytosines. So that's what's going to happen during the synthesis component of interphase during mitosis. So there's your parent molecule, there's their separation, and then there's the daughter nucleotides coming in. We call this a semi-conservative model of replication because each of the initial daughter molecules will contain one of the old strands that were from that parent molecule. But there were a couple other models that were in consideration at the time. One was that although you might have used the parent strands to create the daughter strands went together and the parent strands went together. So that's considered to be the conservative model. And the dispersive model is a mix of the two. Meselson and Stahl were able to do some experimentation that determined that the semi-conservative model was the most logical fit. They used radioactive isotopes again to label nucleotides. The original parent strand was labeled with a heavier isotope of nitrogen, and then the subsequent strands that were formed, their nucleotides were labeled with a lighter isotope. What was seen through the first replication was there was just a single band of hybrid DNA, so you weren't keeping the parent strand together, and so that knocks out the conservative approach. The second replication produced light DNA as well as hybrid DNA, so that kind of knocks out your dispersive model. So there you can kind of see you had your original DNA with the heavy isotope, and then after it was replicated, you had one band, showing that you had a blend of that heavy isotope with the lighter isotope. With the second replication, because now you're taking each of those strands and replicating, you've got two strands that are made solely from that lighter isotope and two strands that are blended. So you're going to have about a 50-50 split between those bands. With the dispersive model, you would have seen more of the same band size because they would have been a blend of one another. You would not have seen just the two separate bands that we see with the semi-conservative. So replication is pretty amazing given how fast it happens and how accurate it has to be. There are lots and lots of enzymes and proteins involved in the replication process. We are definitely not going to hit all of them. We're going to hit a few. Replication begins at these sites known as origins of replication. The DNA strand opens up and starts a bubble. Eukaryotic chromosomes, because they are super duper long, have lots of these origins of replication, and they continue until the molecule is completely copied. With your prokaryotic cells here at the E. coli cell, because you have that circular strand of DNA. It's much smaller. You just need that one replication bubble, and you just allow it to continue in both directions until you have your two bubbles. You have your two circular strands of DNA. You can see in that picture, that microscope picture at the bottom. Micrograph, I think, is the word for it. I just wasn't coming up with it right there. You can see the different origins that are marked there with the pink. So when replication occurs, at the end of each replication bubble, you have this replication fork, and that's where the DNA strands are growing. They're elongating. Helicases help to unwind the double helix at that fork so that the DNA is available to be replicated. DNA does not want to be single-stranded, so SSBPs, single-stranded binding proteins, will bind to the single-stranded DNA and help to stabilize it. And then topoisomerase will help to correct some overwinding of the replication forks so that it's able to not break the helix that's present with your DNA. It will prevent overturns. So it says it can break, swivel, and rejoin the DNA strands. If they have swiveled too far, that's when it would break them to try to get them to rejoin properly. So it kind of overcorrects for if you've gone too far with things. I kind of liken that one to in driver's ed when you've got your teacher in the car with you, and if you haven't done what you need to and there's a possibility of an accident, they might take over. So we also are going to talk a little bit about primase, but there you can kind of see the topoisomerase and your helicase opening up that fork and then the single-stranded binding proteins, and you see them on both sides of the DNA strands, kind of helping to make them a little more stable. So primase, I told you we'd come back to this. Polymerases, DNA polymerases, are what are going to add nucleotides to create those daughter strands of DNA. But they can't add they can't add nucleotides initially. They can only add them to a 3' end. And so RNA primers are going to help to make that possible. Because remember, when you open them up, there is nothing on that initial side of the strand. So primase will help to start a small RNA chain. And why we want it to be RNA, it's going to give that DNA polymerase a chance to add on the DNA nucleotides. And because it is RNA that is there that's getting things started, remember you can't start with DNA. You will use another enzyme that will come along and replace those RNA nucleotides with DNA nucleotides so that the DNA strand is replicated completely. We'll talk about a couple of DNA polymerases. There are lots of DNA polymerases, but the DNA polymerases are able to elongate your DNA. They catalyze that process at your replication fork. We talked about why they need the primer, and then you have to have the DNA strand to know which bases to pair up. For bacteria, you can do around 500 nucleotides a second. For human cells, it's around 50. The nucleotides are basically a nucleoside with phosphates, so it's similar to GTP and ATP. The difference is a sugar. And when they join, two phosphates leave, and you are left with a single phosphate, which is what we find in every one of our nucleotides. So because you have anti-parallel strands and you can only add to the free 3' end where that hydroxyl group is available on your deoxyribose sugar, your DNA strand will only be able to grow from 5' to 3'. So you have two strands moving in opposite directions. The one that is moving towards the fork is known as your leading strand. And DNA polymerase 3, DNA Pol 3, is able to add bases to that strand to generate the daughter strand continuously. To elongate your other strand, because you're working away from that replication fork, we know that as the lagging strand, and so it gets added in chunks. There are lots of RNA primases put at a lot of places to add on that RNA primer, and basically the bases are added up to that RNA primer. DNA polymerase 1 is able to go in, again, because you can't start with DNA Pol 3, to add on bases. But you need to replace that RNA primer. So DNA polymerase 1 will break down those RNA nucleotides and replace it with the DNA nucleotides once you've already got the majority of your fragments complete. Those fragments are known as Okazaki fragments, and because they are synthesized kind of independently of one another, they get joined together with DNA ligase. So again, up here at the top, you started with your RNA primer to get things started to add on to 3', and you can see that that goes along continuously. Over here, because you're going from 5' to 3', you're working away from the fork. And so you can add on a little bit of that primer, but you have to do it in lots of places to be able to get polymerase to add on nucleotides up to those RNA primers. So because it's going kind of in that opposite direction, it's more open-ended, it can only do it in smaller chunks. All these proteins that are participating in DNA replication are part of what we call the DNA replication machine. It could be stationary, but it's also thought, and I know I showed this to you in one of the videos from class, that it could actually reel in those DNA parental molecules and then push out the daughter molecules that are formed. So with all this replication coming on, we need to have a double check in place. And so we do have some enzymes that are able to proofread and handle any repairs with DNA. When you have a mismatch, you have incorrectly paired a base. Maybe you try to pair an A with a G or a C with a T. And so there are some repair enzymes like nucleases, which are able to correct for those errors. You can also have damage to DNA due to environmental effects such as chemicals or physical agents, cigarette smoke or X-ray, and sometimes DNA can just undergo inadvertent changes without there being any sort of environmental issue. Nucleases are able to help to cut out and replace stretches of DNA that are not working properly, that are damaged. And so when those are replaced, then you have DNA polymerase come in and fill them in with the correct nucleotides, and DNA ligase seals it all together. So these altered DNA nucleotides, the error rate once you have proofreading take place is pretty low, but it is not zero. And because this DNA, especially as a result of what's found in your gametes, gets passed on to your offspring, those sequence changes can be permanent. So mutations are the source of genetic variation that allows natural selection to operate. When we are using DNA polymerase to copy our DNA, there are some limitations to it, especially with linear chromosomes, because we can't complete those 5' ends since you can't add bases. You go from 5 to 3, but you don't ever add bases to that 5' side. So when you continue to replicate, because you're doing these anti-parallel strands, you're going to cause your DNA molecule to have uneven ends and it's basically going to shorten. For prokaryotes, not such a big deal because they're circular. And so eukaryotic chromosomes, their DNA molecules have these nucleotide sequences at the end known as telomeres. Telomeres don't have any impact on the shortening process, but they are not coding for genes of consequence. And so they do at least temporarily postpone, hopefully for the long term postpone, any gene erosion that would occur at the end of your DNA molecules. And one hypothesis for this telomeric shortening is that it's contributing to the aging process. So there you see your leading and your lagging strands. When it comes to your lagging strand, you've added that primer on, and at that point, when you replace the terminal end of the primer, now you would be trying to add nucleotides on where you have the phosphate group, and you can't do that. So it does cause a piece of that parental strand not to get paired up, to get shortened. And that picture over there on the left-hand side, the orange dots are your telomeres at the ends of your chromosome. And as you continue to replicate, you continue to run into that process. So having the telomeres there, having that telomeric sequence there is really important to making sure that we hold on to the genes that are needed as long as possible. Um, so if you did let them continue to shorten, eventually you would lose genes that are really important. Telomerase is an enzyme, and it's able to help to protect those gametes and those germ cell chromosomes by lengthening the telomere regions on them. And so by having those there, it's going to help to make them more protected for the replication process that they still have yet to go through. If telomeres are shortened, it could protect cells from cancerous growth by basically limiting how many cell divisions you can undergo. But there is some evidence that telomerase will be, because when you are undergoing cell divisions from cancer, you keep replicating and replicating. So your DNA is going to get shorter and shorter. But there has been, because of this evidence of the telomerase activity, it's allowing those chromosomes to continue to add on to their sequence, which would then allow those chromosomes to be replicated and those cancer cells to continue to grow. So chromosomes are not just DNA, they're packed with protein. With a bacterial chromosome, we've talked about how it's circular, it's double-stranded, it's got a little bit of protein. Eukaryotic chromosomes are linear and they have lots of protein. In bacterium, that DNA is supercoiled and it's found in a certain region in the cytoplasm of that bacterial cell known as the nucleoid. While chromatin, we talked about that a lot with cell division, is not just DNA, it's also protein, and it's what's found in the nucleus of your eukaryotic cells. Chromosomes are able to fit into the nucleus despite their relatively extended length through a pretty involved packing system. And so we'll see, there you see your double helix, you see the histones, and then you see the DNA being wrapped around those histones or groups of histones known as a nucleosome. And then you can wrap the nucleosomes around one another to make fibers. Those fibers can form loops, and then they can scaffold on one another, and we get our chromosome. And you can see there, because you have the replicated chromosome, we've gone from having a 2 nanometer double helix all the way to a 1400 nanometer replicated chromosome. So chromatin is able to undergo packing changes during your cell cycle. In interphase, you have some of it packed into a 10 nanometer fiber, but then it gets compacted into a 30 nanometer fiber via lots of folding and looping. Although the chromosomes are not highly condensed in interphase, they are typically found in certain regions within your nucleus. It does become more loosely packed during interphase and then condenses as prior to mitosis. When it is loosely packed, it is known as euchromatin. When it is highly condensed, when the chromatin and the centromeres and the telomeres are highly condensed, you have heterochromatin. When that is taking place, the dense packing of it makes it difficult for any sort of transcription process to occur, for the genetic information to be expressed. And so histones can cause, when they are modified, can cause changes in how the chromatome is organized. So there you see different colors representing your different chromosomes, and there you see specific locations. You can see from the colors where you're likely to find those chromosomes at in the nucleus.